Okay, so this is the continuation of the exam 2 review. The construction people are gone, and so it's a little more quiet. Okay, so I'm going to share. So we did chapter 6 and 7 previously, and this is going to be 8, and then we'll do 9, and that'll be all the material for the test. So chapter 8 is about metabolism. Remember, metabolism is the totality of all of the energy transformations that occur in your body. That includes ana, which means build things up. So we learned in chapter 5 that if we link, say, glucoses together, we can build bigger molecules called macromolecules. That would be considered an anabolic process because you're taking small molecules and building them up. Taking amino acids and linking them together to make a big protein, that's anabolic. Metabolism is breaking things down. So if you took amylose and stuck in your mouth and broke it down to maltose, which is a disaccharide, and then that broke down in your stomach into glucose, and then underwent glycolysis, and all this breaking down big molecules into small molecules is called catabolism. So metabolism in the mainstream, people think about breaking stuff down, like she can eat whatever she wants, she's got a fast metabolism. That's not really true. She would have a fast catabolism, which means that she broke down food very quickly. Or, that guy can bulk up really fast. He's got a fast anabolism. All right. So we just talked about that. Make sure you know the definition of metabolism, that these things in orange that are highlighted are important. Metabolic pathways are the series of enzymatic reactions. Make sure you know what an enzyme is, what it's made out of. It's a protein. We talked about catabolic. It's breaking down molecules. That usually releases energy. Think about digestion. Anabolic is building molecules like anabolic steroids. They require energy, right? You got to go to the gym if you want to get big. And then these are just the base roots for that. So, and we can break down carbs, lipids, and proteins. All you got to do is look at a nutritional label and you'll know that. We can also build up carbs, lipids, and proteins. So if you sat around and ate a bunch of carbs, you'd make fat cells, for example. This is the all of the different points here symbolize enzymes and different reactions. This is the majority of your metabolic processes. And like I said, this big long chain is the one that we talked about in glycolysis in chapter 9. This is the Krebs cycle right here. And then remember, all those things go to the electron transport chain. So all of these are enzymes. They're like workers in a factory, and it's kind of like a assembly line. So each worker has a different job. Let's say that they're building a car. This person might put in the carburetor, the tires, the steering wheel, the hood, and so on. All enzymes are real specific for what they do. They only do one job, and that's it. And they don't become part of the reaction, right? They're just workers. You're not, just because you're building the car doesn't mean you can become part of the car. All right, we talked about energy. Make sure you know the difference between kinetic and potential energy. Remember, kinetic energy is motion. Anything in motion is kinetic energy. Your car driving down the street, you running down a track, the motion of molecules in the form of heat, the energy that goes from one place to another in the form of light, all those are kinetic. Potential energy is energy that's stored. Think of it as like a battery or a Snickers bar. So it's usually chemical energy, but it could be something else like a piano tied to the top of a building would have potential energy too. It's not moving, but when you drop it, it can do damage and work. Okay, so we can convert energy from one form to another. No big thing there. I'm sure you know this. Gasoline is chemical energy. You put it in the car, that gas gets burned and causes your car to go forward. So the potential energy in the gas is converted to kinetic energy of your car moving forward. And your body does the same thing. We talked about spontaneous reactions. These are reactions that go on their own. You can think of a spontaneous reaction as like once you start gasoline burning, it goes to completion. You don't have to do anything. You don't have to put any external energy in. That's a spontaneous reaction. It once it starts, it continues to go. A non-spontaneous reaction would be something that wouldn't uh, be the reaction wouldn't occur without some sort of outside influence. So unless you heated up the gas or put a match to it, it's not going to start burning. But once it does, it's spontaneous. So non-burning gas would be non-spontaneous, burning gas would be spontaneous. And we can convert non-spontaneous reactions into spontaneous reactions. Throw a match in a gas can. You just converted a non-spontaneous reaction to a spontaneous reaction. We could explain this mathematically through thermodynamics. I don't care that you know the numbers behind it or whatever. You know, there's a whole branch of science that people do that study this. You know, this is how they figure out how to make cars more energy efficient, how to make solar panels more energy efficient, and stuff like that. So, you know, if you're into that, then you can get a job doing this. Engineers do this generally, but chemists do it too. So we're going to talk about chemical reactions, not the movement of cars or whatever, specifically in biology, because that's this class. Make sure you know the first law of thermodynamics, which means that energy can be transferred or transformed, but it can never be created or destroyed. That means all the energy that is in the universe has always been there and always will be there. You can't make it and you can't get rid of it. This is one of the most tried and true laws of all of science. Second law of thermodynamics is every energy transformation makes the universe more disordered. So you're always going to lose a little bit of heat in this, and that heat radiates into outer space and then just dissipates. So it's basically lost energy. That's how hybrid cars work, and even electric cars, they use regenerative braking. Normally, when you brake a car, the brake pads would heat up and you would lose that heat energy out into the universe. But some, smart engineers figured out that studied thermodynamics that you could take that heat energy from braking and then convert that back into the chemical energy of electricity, and so that you can make your car go further. So that's basically how that works. And remember that these terms, entropy is a measure of disorder in a system. So tell me you're not laughing at me. Okay. All right. So we already talked about spontaneous. So I'm not going to go over it again. Make sure you know what enthalpy is. Enthalpy is the energy. When we're talking about chemical reactions, it's the energy in the chemical bonds. Entropy is disordered. Enthalpy is symbolized by H. Entropy is symbolized by S. I will give you these H's and S's. I will give you the equation, but you need to know these words, these terms, and you need to know what they symbolize, and you need to know what they mean. So make sure you know the definitions of that. I think it's in the crosswords. Okay, we talked about entropy. I will give you this equation on the test, but you need to know what this means. So you need to know what G is, Gibbs free energy. This is enthalpy, the energy in the chemical bonds, temperature, and then disorder, which is entropy. So you need to know what all of those mean. You should know that this delta means change. All right. So in order for a non-spontaneous reaction to become spontaneous, this has to be negative. We can make it negative by reducing the energy in the bonds, or increasing, because this is a negative, increasing the temperature or increasing disorder. So you should know that. You should also know that in living systems, we cannot use disorder because that goes against our rule that we did in chapter 1. And we can't increase temperature because that changes the shape of molecules and would kill you. So the only thing that living systems can do to get this to go down is to reduce the energy in the bonds. And it does that by adding a phosphate, which changes the shape, just like adding a Lego block to a Lego tower changes its shape, and that shape change is like bending a stick. It weakens the bond so that you can break it more easily. All right. So we can make delta G go down by increasing temperature, which is what I said, reducing the energy of the bonds or increasing disorder, but we can't do T or S because in living systems. Scientists love to get things outside the body because then we can use T and S. Like, for example, if I wanted to copy DNA to find out who the baby's daddy is, I would take the DNA out of the cell because I can use temperature to manipulate the chemical bonds of DNA to open that book, so to speak, where I can only do that by changing the energy of the bonds by adding phosphates to it inside the body. And that's not efficient and it's not very easy to do. So scientists, biologists like to take things out of the cell to study it because then we can use these other ways to get reactions to occur. Okay. So we have exergonic, that means exo is out, gonic is energy. This gives off energy. That means the energy in the reactants is higher than the products. Remember that we can't create or destroy energy, we can only change form. So the difference in the energy in the reactants of the products have to go somewhere. And in this case, it can be output as heat. It could be output as chemical energy in the form of ATP. It could be output in many different ways. But we don't use the word exothermic because that implies heat is the only form of energy that you can use, and that's not true. We know living systems can make ATP. So we say outside, it gives off energy instead of gives off heat. And just, you know, when I'll put this on the test and I might ask you, you know, what kind of reaction is this? Is this an exergonic or an endergonic? You would say exergonic because rocks roll downhill, right? Reactants have more energy than products. You should know delta G is negative in this, right? It's a spontaneous reaction. Rocks spontaneously roll down the hill, and that's a negative delta G. You don't need to know these numbers. I've just given you a value so that we can do some clever math later. Endergonic, remember that requires energy. We have to put energy in because the reactants have less energy than the products. So let me just back up. A good example of this is what you do. So the reactants, sugar and oxygen that you're taking in, have more energy than the products. Otherwise, why would you eat and breathe air? You wouldn't need to because you wouldn't get anything out of it. So respiration is a good example of this. That's an exergonic reaction. You get energy out of it. Photosynthesis is a good example of endergonic reaction. The reactants, which are carbon dioxide, that's what we exhale, right? Those are the products here. In photosynthesis, the reactants are the products from respiration. So carbon dioxide and water, and the products are glucose and oxygen. These have a lot more energy than the reactants. So because of that law of thermodynamics, we have to input an equal amount of energy to create these products. And that energy can come from lots of places, but in photosynthesis, it comes from sunlight. Remember, this is a non-spontaneous reaction. Rocks don't roll uphill. This is just showing you that we have to put in the same amount of energy to create a mole of glucose as we could get out of a mole of glucose. And that life is an open system, like this artist is showing. If the water above our dam and below our dam are equal, it's not going to turn the turbine and we won't get any energy. So delta G is 0. We don't want delta G 0. It has to be greater than or less than 0 to make an unequal reaction here to create energy, or to use energy to create other things. And to do that, we can open it up, right? And so that's why the environment's important. This really isn't an environmental biology class, but that's why. So we talked about ATP. We talked about it's a nucleotide. Remember, it's closely related to RNA. I might ask you that on the test. It's unstable, so it's like a compressed spring because of the negative charges on it. And we can use this to do a lot of things. We can do it to use it to do mechanical things like shape changes. We can transport, like we talked about with the sodium potassium pump. We could do chemical reactions of weakening bonds by adding phosphates. These are the negative charges that make it unstable. Remember, we have to add water to break that bond still. It's hydrolysis, and we release energy. And we end up with ADP. That ADP is recycled. This is an example of how we can add a phosphate. So phosphorylation is adding a phosphate to glutamic acid to produce glutamine, which is an essential amino acid. So this reaction would never occur without the presence of ATP. We add the phosphate that has that negative 7.3 kilocalories, and so we've taken a delta G that was positive and turned it into a delta G as negative, and that's a spontaneous reaction. That occurs inside your body all the time. You have to regenerate ATP. There's no way you could consume the massive amount of ATP you burn through in a day, about 150 to 180 pounds, depending on your body weight, and the amount of energy you consume or use, sorry. So anyway, there's no way anyone can eat 150 pounds or 180 pounds of ATP that they need. So your body has to recycle it. It simply does that by adding a phosphate. So it's diphosphate and it becomes triphosphate. And in chapter 9, we talk about how this is done. We talked about enzymes. Remember, they're catalysts, they're proteins, they accelerate a reaction, but they don't become part of it themselves. All enzymes are catalysts, but not all catalysts are enzymes. They're chemical catalysts, like in your car, there's a catalytic converter. It's not a protein, but an enzyme is a catalyst that is a protein always. It reduces the activation energy. And I promise you that this drawing will be on the test, and I'm going to ask you, what is this? What is this? These arrows represent, and it won't be on there. So make sure you know transition state, reactants, products, energy of activation, which is this hump, where we have to put in some energy to get the rock to start rolling down the hill. And then delta G is simply the difference of the energy in the reactants and the products. And then transition state is when the state, remember, we've got to break these bonds to form new ones. So here A and B and C and D are bonds. We're going to break those and then make new bonds. That's really all a chemical reaction is. Enzymes speed up reactions by lowering that activation energy. They don't change delta G. Remember, that's really important. Enzymes do not change delta G. So if I ask you on the test if enzymes change delta G, you better say no, or you're going to get it wrong. Enzymes are real specific for the substrate. So substrate is something an enzyme acts on. Enzymes end in ASE, so lactase would react with lactose. Sucrase would react with sucrose, and so on. And it really depends on that three-dimensional shape of the enzyme, which we talked about as a protein. And we talked about in chapter 5 how the 3D shape of proteins are made. You should know what an active site is. That's the region where the enzyme binds to the substrate and causes the reaction to occur. Remember, you should also know that this is induced fit, which means that when we add a molecule to another molecule, it changes its shape. This molecule here is hexokinase. A kinase, remember I asked you guys to know that for the test, so I'll ask you on the test. Kinase is anything that adds a phosphate to another molecule. So this hexokinase is going to add a phosphate to this hexose, which is glucose. It's a 6-carbon sugar. Kind of, you can think of it as a lock and key. This is just an example of it. Remember, if you want your reaction to go faster, if you want to break down more substrate, then you need more enzyme. So just like if you want to make more cars, you need to hire more workers. But not only that, you're going to need to increase the amount of parts you have to build those cars. So the amount of substrate is important, right? The amount of parts and the amount of workers is also important. So you can overcome this by hiring more workers, but you also have to increase the amount of substrate. Otherwise, it doesn't matter. Let's say you have 10 workers and they can build 10 cars a day. If you hired 20 workers and you didn't have any more parts than to build 10 cars a day, you'd only get 10 cars. So you have to increase the substrate too. Remember that every enzyme has an optimal temperature and an optimal pH. So an organism that lives in a hot spring, like a heat-tolerant bacteria, this is in Celsius. So this is about 200 degrees Fahrenheit. This is 37, which is about 98.6. So, you would expect the enzymes inside your body to work at body temperature. And, I might ask you, okay, crabs at the bottom of the ocean, what do you think their optimal temperature for their enzymes are? And since you know from chapter 3 that the temperature at the bottom of the ocean is 4 degrees Celsius, you should be able to say that optimal temperature is 4 degrees Celsius. pH is important too. So let's go back to temperature. If we decrease the temperature, the reaction rate goes down. If we increase the temperature, the reaction rate goes down. So that's why it has an optimal temperature. And same thing with pH because of the shape. So this is from your stomach, pH is about 2 in your stomach. This is from your mouth, pH is about 8. So these enzymes are going to work best in whatever pH they're used to and whatever temperature they're used to. Cofactors are small inorganic molecules. I'm going to ask you this on the test, so make sure you know this. Cofactors are inorganic molecules like zinc, copper. Inorganic means anything that's not a carbon that is required to change that enzyme shape to make it work. So without the cofactor, the enzyme is worthless. Same thing when they're vitamins, we call them coenzymes. And vitamins are organic generally, so they have carbon. Like that's why sailors would get scurvy because their enzymes wouldn't work because they didn't have the vitamin C to allow them to work. So that can cause all kinds of issues if you don't have these trace elements. That's why, people sometimes supplement because they don't get these from their diet. And they're super important for your body to function. We can have things that block the active site, so a competitive inhibitor. We can have something somewhere away from the active site, like a non-competitive inhibitor that would change the shape so the substrate can no longer bind. So that's things that can affect enzyme activity. This happens all the time. Your body does this constantly. We talked about allosteric regulation. You can have an activator. We talked about like lactose activates the enzyme to break down the sugar in milk, lactase. And then we could have an inhibitor. Like if you have tryptophan, you don't want to make anymore, so it shuts it down. You could have multiple enzymes with active sites functioning on different sides of the protein, and we saw this in chapter 9 with ATP synthase. It has 3 functional sides on that one enzyme. We talked about feedback inhibition. You can use the product to shut down a pathway. In this case, threonine is made into isoleucine. But if you have enough isoleucine, then you don't need to turn threonine into isoleucine. So isoleucine binds to this, changes its active site so it doesn't work anymore, and shuts down this whole pathway. So that's an example of feedback inhibition. All right, that's chapter 8. And then so here's chapter 9. Let's go back. So in chapter 9, I think this is still sharing. Should be. It's highlighted in green. So anyway, if it's not, I apologize. Anyway, so we talked about phosphates. ATP donates phosphates and makes ADP, which is shown here. We can use it to change the shape of a channel, like the sodium-potassium pump. We can use it to change the shape of a molecule to make it walk, like in the motor proteins we talked about in chapter 5. We can use it to decrease Gibbs-free energy, so we can make a non-spontaneous reaction spontaneous, which we talked about in chapter 8. So we can, ATP can do all of these things. It's really important. And you make ATP in 2 ways: fermentation, which is the absence of oxygen, or cellular respiration is with oxygen. Fermentation is anaerobic, which means doesn't require oxygen, and we partially degrade sugars, the glucose, into pyruvate. It only produces 2 ATP. It's not enough for you to live. You want to test that, hold your breath, see how long you make it. Because without oxygen, you undergo fermentation. In respiration, we make a lot more. And respiration occurs in the presence of oxygen. So we break down glucose into energy, which is ATP, and we give off carbon dioxide and water. Make sure you know what a redox reaction is and where electrons get transferred. I went over this, so you should know that NAD is an electron carrier, and that becomes NADH, and its sole job is to safely carry electrons to the electron transport chain. You should know that this looks like a nucleotide because it's 2 nucleotides linked together that we talked about in chapter 5. You should know that the enzyme that does this is called a dehydrogenase. D means to take away hydrogen, and then ACE means it's an enzyme. So it's taking away hydrogen off of glucose and putting it on this NAD plus to make NADH. So in this case, it's an oxidation reduction reaction. Glucose is losing electrons. So when you lose an electron, you're oxidized, and then NAD plus is gaining electrons. So when you gain electrons, you're reduced. So glucose is being oxidized, NAD plus is being reduced to NADH. Three steps: glycolysis, Krebs cycle, electron transport. Remember, Krebs is also known as the citric acid cycle. You should know both. This is a summary slide. I'm going to ask you where glycolysis occurs, like I said, in the cytoplasm. It makes electrons carried by NADH that gets put in the electron transport chain. Glucose is made into pyruvate. This occurs in the cytoplasm. We make 2 ATP here. We require these 2 ATP to actively transport pyruvate into the mitochondria. That makes acetyl-CoA. So our net ATP is 0. In the Krebs cycle, we make 2 ATP, and then all of the other ATP is made in the electron transport chain from the electron carriers, NADH and FADH2. So glycolysis is catabolic. That means it's breaking down glucose, splits it into 2,3 carbon sugars. They get oxidized. So if it's oxidized, then it loses an electron. Remember, like glucose loses the electrons. What gains the electrons is NAD plus or FADH plus. Both of those gain electrons from glucose. Reaction is catalyzed by enzymes like everything else inside your body. This occurs whether or not oxygen is present. So glycolysis happens. We have energy investment, so we have to pay 2 ATP. We get 4, net is 2 NADH, 2 pyruvate. Remember, I told you you need to know what goes in and what comes out. So for each step, glycolysis, glucose goes in, 2 ATP go in, comes out 4 ATP, 2 NADH, 2 pyruvates. That's all you need to know for this. That's all I'm going to ask you. You don't need to know all this. I'm just showing you this is how it works on a chemical level. You do need to know what a kinase is. I'm going to ask you that. I don't care that you know this. Like I said, I want you to know what goes in and what comes out. So what is being put out of this is important. Pyruvates, ATPs, NADHs. Again, this is pyruvate goes into the mitochondria. It turns into acetyl-CoA. We make NADH and carbon dioxide. So again, we said what goes in: 2 pyruvate, 2 ATP. What comes out: 2 CO2, 2 NADH, 2 acetyl-CoA. And then this is right now we have 4 NADH. So we have 4 electron carriers, each carrying 2 electrons. Krebs acetyl-CoA goes into here. This is called the citric acid cycle because it converts oxaloacetate into citrate. You don't need to know all this. If you take biochemistry, you will. But like I said, you need to know what goes in. 2 acetyl-CoA go in. And what comes out: 6 NADH, 4 CO2, 2 ATP, 2 FADH2. And that's shown here. Remember, there's 2 of these, so you have to multiply each of these by 2. So right now we have 10 NADH and 2 FADH2s and 2 ATPs. You should be able to know what the summary is for each one of these steps. So this is the 3rd step. Step 1 is glycolysis. Step 2 is converting pyruvate to acetyl-CoA. Step 3 is the Krebs or citric acid cycle. And then step 4, and this is just a summary, is the electron transport chain. So here's where all the electrons come off. I told you the story behind it. You don't really need to know the story. I just did it so that you do need to know the order of these complexes. So I highlighted them here in yellow. You need to know complex 1, complex 2, CoQ, complex 3, cytochrome C, and complex 4. You don't need to know any of this. You need to know the electrons go from complex 1 using NADH to, they skip complex 2, they go to CoQ, to 3, to C, and to 4. So they're going through 3 integral membrane proteins, pumping 2 protons out of each. So 2, 4, 6 protons are pumped. That produces 3 ATP. So every NADH makes 3 ATP. You need to know the path FADH2 follows. So complex 2, CoQ, complex 3, complex C, site C, and complex 4. Remember, this is an integral membrane protein, complex 3. This is an integral membrane protein. So the 2 electrons pump 2 protons here, 2 protons here. So 4 protons. Remember, 2 protons give us an ATP. So we get 4 ATP for every FADH2. This is just the order, but you only need to know these, like we talked about. This is just kind of showing you how it works. You need to know who Peter Mitchell is. You need to know that we're doing this to make an electrical potential. You should remember that if we increase H pluses, we decrease the pH, right? So The more hydrogens you add to a solution, the lower the pH. That's how they proved that this was real. Won a Nobel Prize. Should know what an elementary particle is. Should know what ATP synthase is. Need to know where F0 is located in the intermembrane and F1 is in the matrix. You need to know that protons power this thing and make it spin, and it takes ADP and phosphate and crushes them together to make ATP. Remember, I just did the math. So we have 10 NADHs. Each one of those make 3 ATP. So 30 ATP here. 2 FADHs, they each make 2 because they only go through 2 integral membrane proteins, complex 3 and 4. So that's 30 plus 4 is 34 from the electron carriers, and then 2 from the Krebs cycle, which is 36 total. So 34 from electron transport, 2 from Krebs. There's going to be a series of questions like this, so make sure you're able to do the math. Why we make 34 ATP, why we make 36 total. This kind of summarizes it. We talked about bacteria can make more. Why that is, I might ask you on the test. Why is that? You might want to look at the lecture if you're not sure. We did a little math to show you that your body is somewhere between 40 and 80% efficient, which is really super efficient. And then these are the questions and things that you should think about. So I answered all of these in lectures, so make sure you guys know that. You also need to know the difference between alcohol fermentation and lactic acid fermentation. You don't need to know all this stuff, but what you should know is what goes in, glucose, and what comes out, ethanol. And what goes in, also NADH comes out and ATP comes out. Again, glucose goes in, ATP comes out, NADH comes out. It's used to make lactate. So what goes in and what comes out. Examples, you should know, you should be able to give me examples of what does alcohol and what does lactic acid fermentation. You don't need to know this whole thing, but what you shouldn't take out of this is that all the stuff that your body uses for energy, proteins, carbs, fats, they're all going through the pathway that we just talked about, just entering in different parts. And that we can run this in reverse to build things too. So not just break them down, but to produce them. That's it. So if you guys have any questions, I'm going to have office hours on Monday. And then your exam was due Monday by midnight. So make sure you start it early. Good luck.